DNA is the genetic material, the molecule that passes information from one generation to the next. We know that now, but how do we figure that out? Hi biology friends, it's Sarah, your other biology professor. Let's talk about the Hershey-Chase experiment. So by the middle part of the last century, some scientists were starting to warm up to the idea that DNA could be the molecule of inheritance. And this was thanks to experiments like those by Griffith, who showed that the genetic material, whatever it was capable of being transferred between bacteria, transforming them from one type to another. And experiments like Avery, MacLeod, and McCarthy's, which suggested that this transforming material was DNA rather than protein. But not everyone was convinced. In fact, most scientists at the time still held the belief that protein would be the genetic material. After all, the alphabet of protein has 20 letters compared to the much smaller alphabet of DNA with just 4. With all that additional complexity we could get with proteins, surely it would be the molecule of inheritance, right? Enter Hershey and Chase. So how were Alfred Hershey and Martha Chase able to address the question as to which of these macromolecules is the genetic material? Well, instead of bacteria, they focused in on the bacteriophage, a type of virus that infects bacteria. Bacteria are cells, obviously, and like all cells, they've got a lot going on, whereas the humble bacteriophage is a lot simpler. This guy, it's just protein and nucleic acid. One of those is the genetic material. During infection, either the protein or the DNA is injected into the bacterium and then replicated in order to make more phages, while the rest of the virus stays outside the cell. So which one's the genetic material, protein or DNA? To answer that question, Hershey and Chase used 2 critical tools. First, they needed a way to separately label the phage DNA and the phage protein so that after infection occurred, they could say, okay, where's the DNA? Where's the protein? Which one actually entered the cell? And for that, they used radioactive isotopes. You may remember, hopefully, that isotopes are atoms that vary in the number of neutrons that they have. So for example, sulfur has 16 protons, typically 16 neutrons. Add those together for a mass of 32. But an isotope of sulfur with 19 neutrons would have a mass of 35. We'd call this S35 or sulfur 35. Sulfur 35 is radioactive. It's unstable. And like many of us, it's on a journey to becoming something more stable. And along the way, it's flinging off particles that can be detected. All right, so we've got to label proteins, we've got to label DNA. Which isotopes should we use? Well, since sulfur is found in all proteins, but not in DNA, sulfur 35 is a great choice for labeling the proteins. So one batch of bacteriophages is going to have its protein labeled with the sulfur 35 isotope. What about the DNA? Well, since phosphorus is found in all nucleic acids, but not in protein, P32 was used here. So a separate batch of bacteriophages had its DNA labeled with radioactive phosphorus. We've got phages with labeled protein, we've got phages with labeled DNA. It's time to infect some poor little bacteria. So our sulfur 35 phages, these have labeled proteins, right? These are going to be introduced to some E. coli, given time to infect. So they're going to have time to attach and inject their genetic material into these bacteria. Separately, we are going to introduce some P32 labeled phages. These have the labeled DNA, right? To some bacteriophages. Same thing, give them some time to infect. Next, they had to see which material, protein or DNA, was actually injected into the bacteria during infection. And for that, they needed the second critical tool. And this one was another brilliant piece of engineering, a household blender. Yeah, well, brilliance isn't always complicated. They used a blender to gently separate off the parts of the bacteriophage that stayed outside the cell. So that part would be sort of the structural component of the bacteriophage. It's called the phage coat or the ghost. And again, that would stay outside, attached to the cell, whereas the genetic material would have been injected into the cell. So again, that blending process is going to unattach the ghost from the E. coli, because if it didn't actually enter the cell, it's not the genetic material. After blending, they'd separated the E. coli from the viral bits that didn't enter the cell. But now they needed a way to separate those infected bacteria from those little non-infectious viral components, as well as from any viruses in the solution that didn't attach to a cell to infect in the first place. And for that, they used centrifugation. So in a centrifuge, spinning at high speed, larger, denser components, our infected E. coli, will form a pellet, whereas these smaller viral particles, like the ghosts, will remain in the solution called the supernatant. Now, all that's left was to figure out which of our radioactive labels we could detect in the infected bacteria. That's our genetic material. In the batch where the phages were labeled with sulfur 35, so the proteins were labeled, radioactivity was found in the supernatant. So that means that the protein makes up components of the virus that stays outside the cell during infection, not components that are injected during infection. So not looking so good for proteins as the genetic material. And in the batch where the bacteriophages were labeled with P32, the labeled DNA, radioactivity was found in the pellet. That means that the phage DNA, again labeled with P32, was passed from the phage to the bacterium during infection. DNA is the molecule of inheritance. It's our genetic material. Just to tie things up neatly, they also looked at the phages that were replicated inside of the infected E. coli cells. And from those phages, they found labeled DNA, DNA with P32. Again, highlighting that DNA was passed from the phage to the bacterium during infection and used to create the next generation of phages. So Hershey and Chase found that viral DNA, as detected by P32, is transferred from the bacteriophage to the E. coli during infection, while viral protein was not. So this provided very strong evidence that DNA was the genetic material. This work was very convincing to the scientific community and sparked a lot more interest in learning about DNA. In fact, just a year later, the double helix structure of DNA was published. All right, give me a, if you learned something about the Hershey-Chase experiment, subscribe for even more biology tips and videos from me. Check out my website for tutoring resources, and I will see you next time.